Well, yeah. So first of all, thank you to the organizers for inviting me to this interesting symposium. It's really fun and a pleasure to share some of my recent work in this area. So I'm not, not sort of a deep learning person. I'm a microscopist, and I build microscopes to look at biology at different spatiotemporal scales, optical microscopes. And over the years, these have spanned everything from trying to image focal adhesions with live cell palm to other kinds of molecules, cytoskeletal proteins in 2 and 3 dimensions across time, sort of larger length scales, looking at groups of cells and embryos migrating, and then at even larger scales, more recently, I've been sort of dragged into tissue imaging, imaging large sections of cleared tissue at cellular resolution. And when I was thinking about it, the sort of unifying theme across all of these different data sets that I'm showing here is that computation is in fact essential, not just for rendering the images, but also for interpreting the data and for converting the raw data into something that a biologist can analyze. And so today, I just sort of thought I'd talk about some more recent work in this area, which is using deep learning for image restoration. And as a microscopist, I think this is really an interesting playground because there are trade-offs in the physics of microscopes, right? So it's really hard to build microscopes that have fantastic spatial resolution and really can image rapidly and have low light exposure so you don't fry your sample and give high SNR images. These axes of performance are strongly coupled in microscopy. And deep learning kind of caught my eye with this work by Weigert and colleagues a few years ago. There have been other papers in this area as well, but these guys really did a great job of kind of illustrating what could be possible. Using data-driven prior information in a principled way to kind of expand what is possible. And this paper caught my eye, and we started thinking about working in this area ourselves. And I'm always particularly interested in trying to figure out what are the limitations of these techniques. So I'll speak a bit about that in the first half of this talk. Now, really, the workflow for these methods is broadly speaking always the same. So I've illustrated that here in the case of denoising, where you might acquire low signal-to-noise ratio and high signal-to-noise ratio matched pairs or matched volumes of some structure you're interested in. You use these data sets to train a neural network, and then given that trained neural network, you can challenge the neural network with new, let's say, noisy data that the network has never seen before, and then you make a prediction where you try and denoise the data. And our foray into this field is really a collaboration with S-Vision and the Avia team. We co-opted a residual channel attention network framework that was, we didn't invent. It was shown to produce better images of high resolution structure on natural images than alternative CNNs. And we were just sort of curious to know, does this scale to fluorescence microscopy applications? And the S-Vision team developed a 3-dimensional Arcad version that we used on a bunch of different microscopy data sets. And if you're interested in more of the details, you can check out our bioRxiv paper. And if you're interested in trying to use the code yourself, you can download it from this GitHub repository. So we started trying to play with denoising applications. And so in my lab, a number of years ago, we developed a rapid structured illumination microscope, the so-called instant SIM or ISIM. And we used this microscope to start playing around with denoising. So for example, in this set of images, we took raw data at very low SNR of, in this case, mitochondria. The mitochondrial label labels the outer mitochondrial membrane protein TOM20. And then we had ground truth data acquired at something like 100-fold higher illumination intensity. And then we could use these matched pairs to train Arcan, CARE, SSRSNet, and ESRGAN, these kind of state-of-the-art neural network architectures. And on these organelles, we found that Arcan did have sort of an edge. So both qualitatively, we could recover the shapes of these mitochondria better than these other networks, and quantitatively as well. So if you look at sort of volume measures of performance like structural similarity index or peak signal-to-noise ratio, then the Arcan outperformed these other networks. And that sort of held in lysosomes, microtubules, actin, Golgi, and ER. So that was sort of an encouraging sign. And these neural networks were trained on fixed pairs of volumes, but I think the real strength of these neural networks is you can apply them to live data sets as well. So to sort of motivate that, this instant SIM is not a Lightsheet microscope. So it volumetrically doses the specimen throughout the volume, which means that if you look at four-dimensional applications. In the first volume, in this data set over here, where we've transfected cells with a mitochondrial fill, the first volume looks great. When we turn the laser illumination up, you can see all of this subcellular mitochondrial detail. If you play this movie over time, you rapidly bleach the fluorescence, and you can quantify that bleaching in this curve, where in the first 10 volumes, we've extinguished the pool of useful fluorescence. Now, one way of dealing with that, of course, is to turn the laser way down. And if the laser illumination is low enough that the rate of photobleaching is small relative to the rate of protein synthesis, you can image essentially forever for all practical purposes. The problem is that the signal-to-noise ratio at that level of intensity is useless. However, using the Arcad, we can actually restore the signal-to-noise ratio to the point where we can actually segment mitochondria and quantify interactions. And in fact, in this particular example, we were able to record for thousands of volumes or tens of thousands of images without any apparent photobleaching. And one of my colleagues, Phil Lassou, also did a quick look and also didn't find any significant mitochondrial fragmentation in the restored data set. So, to me as an instrument developer, this was kind of amazing, right? I mean, for once, the fluorescence signal actually rose over time, which is something we never saw with these super resolution modalities. But the restoration could give us data we couldn't actually obtain with the base microscope. And this sort of held in other examples. So we could look at a two-color example where we look at mitochondria in green and lysosomes in red. And again, in the raw data, the lysosomal signal is barely visible, but after restoration on trained Arcad that had been trained on fixed lysosomes and mitochondria, we could actually restore data to the point that we could quantify mitochondrial and lysosomal interactions over hundreds of super resolution volumes. So again, this was very encouraging, but it made us wonder, where does the network break down? And so we tried to take a close look at that using another structure, in this case, the ER. And so if you compare the ground truth data taken at high SNR to the Arcad prediction taken with much lower SNR input, you notice a few things. So one point is that, coarsely speaking, the Arcad gets it right. Many of the features in the ground truth are represented well. However, if you look more closely, you find instances where the Arcad, it either makes up something that isn't there, or it gets the structure wrong in a subtle or not so subtle way. And it would be nice to quantify where these failures occur, so you at least know what you're looking at in these predictions. And so we started to take a stab at that. One way of trying to get a handle on this for data is to try and train multiple instances of the Arcad. In this case, for example, 4 different instances of the Arcad with different parameters, and then you can compare networks of measures of network agreement and disagreement. The disagreement could be something simple like the standard deviation in the output of these neural networks, or something more fancy like the so-called ensemble disagreement. But when we did this, we found that these measures of network disagreement were not particularly informative. So you would like to have a measure that sort of correlates or anti-correlates with problems in the prediction of these neural networks, and that's not actually what we found. In fact, what seemed to be better is this sort of empirical signal-to-noise ratio map, where for each pixel in the input data, we just quantified the mean signal divided by some measure of the noise, like the Poisson noise and the read noise of the detector. And that's something where, if we looked closely, if the signal-to-noise ratio dropped below some critical level, that actually did correlate with problems in the network output. So again, just looking at this ER output in sort of a higher magnification view, I've highlighted 2 instances here where the Arcad either made something up or it got it wrong. And if you look at the standard deviation over the mean, the so-called coefficient of variation, or the ensemble disagreement, these measures were not informative. But a good rule of thumb is that if the SNR of the input image is less than about 3, then this is where you can expect some skepticism in the Arcad output. So this is a very crude measure, and what I would say is this result is sort of a plea for help for the people that design neural networks to come up with better measures of disagreement, because this is, I think, what the biologists want. Let me move on to a different application, which is using the Arcad for resolution enhancement. So here, the name of the game is you have some low resolution and high resolution image. You somehow can obtain images of your structure at high and low resolution. You use these pairs or paired volumes, again, to train a neural network. And then you challenge the neural network with blurry input the network has never seen before and ask how well can you deblur. And so to start getting an empirical handle on this, we started with the simulations of small spherical particles that were sort of right at the resolution limit of the microscope of the point spread function. We then took this ground truth and blurred it with a kernel that was twofold larger than the ground truth in X, Y, and Z. And we just asked how well do different neural networks reproduce the ground truth. And what we found is that, at least on this spherical noise-free phantom, all of these networks do a fairly good job. They all, given this blurry input, can do a reasonably good job of reproducing the ground truth. I mean, the SSR, SNET, and ESR GAN are a little bit worse, but not too obviously. The CARE and the Arcad are kind of class leading. And then we started to turn the level of blurriness up. So at threefold level of blur, you can see this input blur is quite a bit more blurry. Now you start to see a breakdown in performance. The SSR, SNET, and ESR GAN give you sort of funny output. The CARE is pretty good, but you can, if you look carefully, it detects fewer particles than the Arcad. And then if you turn up the blur even more, at fourfold blur, now the input data is really blurry, and all networks suffer degradation. The ESR GAN and the SSR net are not ones you would want to use. Even the CARE and Arcad are not good networks. I mean, they miss a lot of the ground truth data. And one can quantify that breakdown in performance, again, using these kind of quantitative metrics like structural similarity and peak signal to noise ratio. So to kind of boil down the take-home message on these phantoms, we found that the Arcad does have an edge, at least on these phantoms compared to these other networks. But the performance of all networks degrades, as you would expect, as you turn the blurring level up. And I think, for me as the microscopist, the kind of, the red flag happens between 3- and 4-fold blurring. So, one way of thinking about this is that for linear structured illumination microscopy, these networks do fairly well with enough training data. But if you go to the nonlinear methods, then you would start to have questions at extreme levels of deblur. So one question is, does this kind of hold on real data? And we were lucky that one of our collaborators, Chris Combs, had a Leica STED microscope. And this microscope had the advantage of acquiring diffraction-limited confocal volumes as well as super resolution STED volumes. So we could acquire diffraction-limited confocal data and STED super resolution data. STED, of course, is this technique invented by Stefan Hell that won the Nobel Prize a couple of years ago. And so then we could use these matched pairs of confocal and STED data to train Arcads and ask how well does the Arcad do? And for different labels like microtubule and nuclear pore complex and DNA labels, what we find is that the Arcad does a fairly good job. We can recover about 2.5-fold resolution improvement over the diffraction-limited confocal data. And if you look at the Arcad prediction, you also denoise the data compared to the confocal or STED data. If you look carefully, there are areas of discrepancy which are highlighted in the nuclear pore complexes. You don't actually recover all of the nuclear pores, but you do considerably better than the confocal data. And so these data sets on fixed cells made us wonder if we could apply the same neural networks on live cell acquisitions acquired in this microscope. One difficult thing in confocal microscopy is to get 4-dimensional data sets, so volumetric time-lapse images of, say, dividing cells. Because again, the confocal microscope is not a Lightsheet microscope, and you blast the entire cell. But if you turn the laser down to the point where the input data is very noisy in resonant confocal mode, now the data is quite noisy, and you can actually observe dividing cells without too much difficulty in 3 dimensions over time. The data are quite noisy. Using the Arcad that we trained on fixed cells, we could get higher resolution data and denoise data. And I think that improvement in resolution is particularly obvious, like as the cells start to come back to interface structure, where you see all of this structure that isn't really there, or it's hidden in the raw input data. This effort also made us wonder if we could improve our instant sim microscope. So in the instant sim, we don't have a ready way of improving resolution. There's no way, unlike in the STED microscope, where we can just turn the resolution so that it gets better. So we wondered if we could use a different modality as ground truth. And so we turned to expansion microscopy. Expansion microscopy is the technique invented by Ed Boyden a couple of years ago. And this is can be thought of as a copy and paste, where you take your fluorescent label on fixed cells and you copy it over to a hydrogel. And then if you add water you can cause the sample to swell more or less isotropically so that now you physically magnify your specimen and can read out the resolution and the structure using a diffraction limited scope. And this is sort of just an example taken in our lab of imaging mitochondria and microtubule networks in a fixed U2OS cell on a confocal microscope. What we found is that the registration challenge to the pre to post that is necessary to train these Arcad and other networks was a major challenge using expansion microscopy, because although you get sort of this quasi-isotropic expansion, if you look at the organelle level, there are subtle disruptions to structure using expansion microscopy. So when we tried to overlay the expanded images onto the instance SIM images, we failed at training good networks because of these local distortions. And so we were able to get around this problem of distortion in the expansion microscopy data sets by synthetically downgrading the expansion microscopy data sets, by, in other words, blurring it and adding noise so that the blurred synthetic data resembled the raw data we acquired with our microscope. In this way, we were able to train neural networks with low resolution data that looked like our instance SIM data, and then of course, the expansion ground truth that we actually acquired on our instance SIM after expansion. And if we look at sort of the output on using this method, if we compare the synthetic input to the expansion microscopy ground truth, we see that the expansion microscopy does indeed provide more resolution as expected. And the Arcad prediction also does better. It's not quite at the level of the expansion microscopy ground truth, but there is this improvement, particularly in the axial dimension on these mitochondrial and microtubule structures. And if you quantify that improvement on actual real data, not synthetic data, we find around a twofold improvement laterally. So you get to around 100 nanometer resolution using this expansion microscopy network, and almost a fourfold improvement in Z. So more of an improvement in Z. And these improvements were enough that we wanted to sort of try using these neural networks on live cell data sets acquired with our structured illumination microscope. And so just to share a few examples, if you look at the microtubule network in a living T cell as it tries to sort of settle on an antibody-coated cover slip, the input data looks okay, but it's particularly blurry in the axial dimension. The Arcad prediction cleans up and deblurs the overall structure, particularly in Z. And again, if you look at mitochondrial, this TOM20 mitochondrial label, if you sort of zoom in on these mitochondria and look at the axial cross-section, in the raw data, it's hard to discern mitochondria, but in the Arcad prediction, it's much easier to do so. So there is this bona fide resolution improvement. So I've given you some flavor of what you can use this Arcad for in denoising and deblurring on commercial microscopes, but you can also use these methods on new kinds of microscopes that an optical engineer might design. So just to give some motivation there, this is unpublished work, but along with ASI, we collaborated with, we built this Multiview confocal microscope where we introduce sharp line confocal illumination through one of three objective lenses that is arrayed around a sample. You can get diffraction limited performance over a several hundred micron field of view. And the advantage of doing this is that, as you may know, in any one view of a confocal microscope, the resolution is anisotropic. It's worse in Z than in XY. But if you could take data from all three views and combine the data using registration and joint deconvolution, you can get sub 300 nanometer resolution in XY and close to 300 nanometer resolution in Z. Another advantage of collecting from three sides is that if you image scattering specimens, confocal microscopy quickly degrades as you go further into the specimen. And just to sort of motivate that problem, here I'm showing you an example on a mouse kidney section, which is of modest thickness, only 35 microns thick. But if you look at the single confocal views in the axial cross-section along this dotted line, life is pretty good near the cover slip, but as you go deeper and deeper in, you get this kind of haze due to scattering in the specimen, and you don't discern a lot of the fine structure of these glomeruli as you go deeper in. However, if you come in from the top two views and you can acquire that data, you can reconstruct a view that more correctly shows all the structure even at the top end of the sample. And this can be enabling. So I sort of obsess about trying to count nuclei in worms. And if you look at this large adult worm, This is something that I would argue it's impossible to accurately count the nuclei using a confocal microscope because of the scattering problem. But by merging these three views and then using actually a third neural network, this mask RCNN with post-processing, we think we can get them all. So we can segment all 2,000 some nuclei, including in the gonad and the brain and in embryos. And this is something where the sample is around 50 microns thick, 50 to 80 microns thick, and the scattering problem is severe on the far side. But of course, no microscopy technique is omniscient. And one issue that I've mentioned before in confocal microscopy is the dose, right? So if you look at a live sample, like this live cardiomyocyte, these are the cells that beat inside your heart. And Chris Combs in NHLBI was kind enough to give us some of these samples that we stained with MitoTracker red. If you look at any individual volume, the triple view result is better than the single view result. The mitochondria are more clearly defined. There is this scattering haze in the raw single view results that are mitigated after you combine all three views. But if you look at a live data set, what you find is that these cardiomyocytes beat. And so this beating, this motion is a problem for serial confocal microscopy. But another problem is you see this front of fluorescence bleaching as we record these volumes over time. So we need methods of extending imaging duration, imaging faster, and using less illumination dose in confocal microscopy. And this is where we could use what we learned from our restoration with Arcad to kind of rescue the performance of these confocal microscopy techniques. So we trained a two-step neural network procedure where we first denoised and then deblurred the data. And I've kind of illustrated that here with this, another sample of worm embryos with a pan-nuclear stain. So if you take data from any one of these sides, you can see that there's a good imaging side nearest the sample and a bad imaging side as you go deeper in. So each of these raw views has this problem. If you combine all three views together, you can jointly deconvolve the data. But at the illumination intensity you need to keep from frying the sample, the reconstruction is kind of noisy. So we can get around this using deep learning if we train one neural network to denoise based on high and low SNR data sets. And then if we take that denoised input, we can improve the signal to noise ratio of our deconvolved reconstruction. So that works nicely. Then we wonder if we could go even one step further. Could we go directly from the raw data collected by just the lower objective and then produce this higher SNR, denoised, descattered data set? And this actually works. So the punchline is you can get the high SNR triple view result using only this single scattered input. And that's maybe easier to see in this cross section and in this volumetric view. So here we're looking at a little later in embryogenesis where we're imaging all of the nuclei, and in the raw data you have sort of a good region and a bad region of performance. The deep learning does a more even job of collecting all of the nuclei. That's maybe more obvious in this cross section where on this far side you just sort of get this hazy mess. And as I play this movie, you can sort of see that sort of the deep learning reconstruction is much better, especially on the far side of this embryo. One advantage of C. elegans is that we know where we are. So in the 1980s, John Sulston got a Nobel Prize essentially for scoring all of these nuclei. So the number of nuclei is exactly known across time. And so we can ask how well are we actually doing. So again, using the mask RCNN to segment all of these nuclei, what we find is the raw confocal data gets about half the nuclei that are there in the worm. If we compare to a dual view Lightsheet microscope we invented in my lab some years ago, we do much better. But the multi-view confocal output from the single view input is even better. We get about 20% more nuclei reconstructed than the dice bib. There's still some room to go. So we're not perfect, but we're much better than the raw confocal input. And so this was very encouraging. One other nice feature of this bespoke microscope is you can do structured illumination microscopy. You can super resolve data on a confocal base. And the reason for that is that you can blank the sharp line illumination. So you can produce images that are like a conventional structured illumination microscope by turning on and off this illumination at high speed. And so you can get 1D super resolution in any one view. Since we have access to 3 views of a specimen, we can improve our overall volumetric resolution to sub 200 nanometer laterally and sub 300 nanometer axially. The trade-off, of course, is that you need more images for the structured illumination mode. You need between 9 to 15 times more images. but we can use deep learning to circumvent this. So we train neural networks to go from the confocal to the structured illumination microscopy mode. And just to show you some output, because I'm running out of time, if you look at the raw confocal input on this actin in a live cell, it sort of looks okay. You can discern dynamics, you can see filopodial movement, you can see the rearward flow of actin, but the multi-view confocal deep learning result is much better. It's much clearer. The resolution is obviously higher, and we're getting this output with 5 to 9 times less dose than classic structured illumination microscopy. So we can image these volumes at high resolution for many tens of volumes. Because we're doing this on a confocal architecture, we can do this sim prediction not just in single cells, but on thicker tissue level specimens. So just to show one example here, if we look at GFP targeted to neurons in the nerve ring region of this anesthetized larval worm, again, if you look at the confocal microscopy input, you can see some structure, you can identify the nerve ring, the brain of the animal, you can see the nerve cords, but you can clean that up a lot and super resolve the output using this deep learning result. So one sort of final application of using deep learning to improve microscopy, we also do a lot of Lightsheet microscopy where we're, again, obsessing about trying to count nuclei. And if we look at pan-nuclear marker in a fairly high NA Lightsheet microscope, then what you see in the Z-stack is that you can discern individual nuclei, but when the nuclei get packed real close together, they're very hard to discern. Even though every nucleus is several microns in diameter, sometimes you get thin cytoplasmic bridges that are separating them that are on the order of 100 nanometers. So any kind of aberration or scattering basically screws you, so that in densely packed regions of nuclei, it's hard for a human or an algorithm to separate the nuclei. Ideally, you would like to de-aberrate, isotropize, and super resolve all of this data. And so that's what we've done. We've trained a sort of a multi-step deep learning pipeline to do this. We synthetically produce aberrations, which we then remove using an Arcad, de-aberrating the data. We then, one advantage of this Lightsheet microscope is we can take multiple views of the specimen and use joint deconvolution to produce a classical reconstruction with isotropic resolution. We can use this to deconvolve the data and make it isotropic. And then finally, using what we learned from expansion microscopy, we've developed methods to expand the worms. And then that gives us a super resolution ground truth. And so we can super resolve the data. So just to show the output of that triple step process, now if you compare these stacks, you can much more easily separate nuclei, even in scattered or densely labeled regions of the worm. You can also see there's all this beautiful subnuclear structure that is just hinted at in the raw data, but we can recover using this super resolution prediction. And this, of course, works over time. So using the Lightsheet microscope, we can image for hundreds or thousands of volumes in writhing embryos, but we can restore this raw data so that the nuclei are better resolved at both between nuclei and within nuclei. So I think this combination of Lightsheet microscopy and restoration may be the most gentle way of attaining super resolution in living, developing organisms or tissue. So just to kind of summarize what I've said, there's been a lot of data, but I think to summarize it all, I think the current paradigm for microscopy is you have your microscope, you take an image or a volumetric image, and if you understand the physics of the microscope, then you can deconvolve the data using an imaging model to improve it. But fundamentally, if the data is not resolved enough, or if it's not acquired fast enough, or if you bleach the sample, you're back to the drawing board and you use a different microscope. I think the great strength of these data-driven deep learning algorithms is that you get one more hook back into the data. The data itself can improve the data. And so you can take data without sacrificing inherent strengths of the underlying microscope. I mean, to me, that's the main advantage of these methods. But of course, there are challenges and opportunities, and the big one for the biologist is how much of what you see can you actually believe? And we've touched upon that here from sort of my perspective in terms of spatial resolution or SNR or linearity. Another practical question I get asked a lot is how much or how little training data is needed for a given application. From an instrument development perspective, I'm interested in building hybrid workflows where I build new microscopes and build new neural networks. And I think the big holy grail for the field is asking, can you bypass the images entirely, right? I mean, the biologist, at the end of the day, doesn't actually care about the image. What they really want to know is how all of the microtubules in a dense EM data set, for example, are are touching each other and connected, or how do nuclei interact and change and split in a time lapse, or how does the brain of an animal form? I mean, and these are all things we infer from the images. So I'm curious, if one can go even further past the images. And of course, there's a big practical bottleneck, which is how do you get these images and these methods in the hands of the users, the biologists? What training programs and hardware need to be in place so that the actual scientist and user can use these methods? And so I'd just like to thank everyone that contributed, both from my lab, University of Maryland, Yale, ASI, and S-Vision. From my lab, particularly Gigi, Pluto, and Jiamin on the neural network side, and Yichong and Min on the microscopy side, and then of course S-Vision, Hideki, Hoyin, and Luciano, and James and Jichu. And thank you.
Thank you, Harry.
Sure.
Love it that you have the MPL jumper there. So thank you so much for the presentation and the update on the latest. Some feedback. I don't know if you need. If you're, I'm just asking everybody, if you're not speaking, you can that it doesn't some feedback. So there is one question already there from Vitaly. You can see it there. iSIM is clearly one of the fastest imaging systems. Bottleneck might be the SCMOS or EMCCD cameras. Both are commercially available. What about more sensitive detectors that are still not commercially available? Any collaborations with material science lab, experimental sensors? So I think this is on the sensing side rather than on the post-processing.
Yeah. So my feeling about this is that camera technology has come a long way in the last decade, and I feel like it's a little bit sort of eking out marginal returns, right? I mean, so there, the latest generation of cameras have slightly lower read noise, and the QE is close to 1. I'm not sure that we'll see much more advanced there, and most of the time we're more limited by the sample than the detector. That being said, I do have a collaboration with somebody who's interested in like super low light flux. So using using detectors that that can amplify even beyond an EMCCD. But I think that application is extremely niche. So sort of like photoluminescence, or cases where you just can't tolerate any degree of light at all. So that's kind of a special application. But I think for most things that folks are interested in, like following organelle dynamics or sort of subcellular dynamics, I'm not sure that we're going to see that much more from better sensors. I think there is some room for faster sensors with sort of same read noise. Generally, what you tend to sacrifice if you go to these cameras that image at hundreds of thousands of frames per second is noise. So there is some room technically perhaps to improve there. But for most of the imaging where you're imaging at maybe 100 frames per second or around there, I'm not sure that there's that much improvement on the sensor side.
Okay. Yeah, I think that probably covers what Vitaly was wondering about. Thank you for that. There is another question here about, I guess, the versatility of Arcan. Can Arcan be used for brain tissue, or for that matter, any other type of tissue?
Yeah, I mean, I think there's nothing necessarily special about the Arcad in that regard. I mean, what I found empirically is probably what most experts know as well, which is that it depends on the structures, right? So I mean, if you have a structure that sort of looks similar to something that you've already trained a network for, then maybe you can get fairly good results by using a pre-trained network. But if the structure in the tissue is very radically different, then you're probably better off retraining the network with good ground truth. But I don't think there's anything sort of particularly special about brain tissue or tissue. It just comes down to the actual structure I think you're trying to look at.
Thank you. I had a question myself. So the last couple of years have been really have changed a lot of the landscape. Before that, if we wanted to do any sort of denoising or restoration, you had essentially point spread function, deconvolution, and little else. How does that compare? And I know you've done some work there, so that I'm kind of want a little bit of know a little bit about that too. So how does deep learning compare in terms of problems that it has that point spread function type deconvolution doesn't, and vice versa, advantages that it may have?
I think for many cases, you can do fairly well with kind of classic techniques. I mean, so the deconvolution that I think you mentioned, it's usually used for improving effective contrast or effective resolution, right? I mean, you can't actually break the resolution barrier, but in really noisy scenarios, if you're not careful with deconvolution, you can end up amplifying noise. And so there, using some of these content-aware methods, or even some of the sort of self-supervised methods can be a boost, right? I mean, you can actually improve the raw data so that you don't amplify artifacts as much and uncover really noisy features that a classic, say, Richardson-Lucy deconvolution fails at. I guess the question is really, what is the application, right? So for many things that we do in our lab, we don't need to use any sort of deep learning. I mean, the classic Richardson-Lucy deconvolution is enough. But for other applications where we can't describe the physics easily with one spatially invariant kernel, which is what is used, that's where we might turn to deep learning in our lab.
Okay, so the more heterogeneous, either heterogeneous samples or harder to model optical. Okay, so there's a couple of other questions, and I wanted to read them out to get a chance for people to get the answer. So one is related to, I guess, the general use of the training data that you have. So how close does the ground state or ground truth data for training need to be to the experimental data? Total intensity, scattering, and so on. If you could comment on that.
I'm afraid I don't have a rigorous answer to that. I mean, I haven't quantified in any really meaningful quantitative way how close it has to be, but sort of as a cautionary tale, I guess, right? So we've trained Arcad neural networks to restore both mitochondrial membrane, like Tom20, and mitochondrial fill. And once somebody in the lab confused the two, and they converted a mitochondrial fill marker into a very convincing Tom20, so that's both a blessing and a curse, right? I mean, on the one hand, it's kind of very valuable, right? I mean, to be able to do this cross-modality prediction, and that's what people have done with bright field recordings, right? Where they take a bright field image and then they train a neural network to restore all kinds of other organelles that have some. But if you're not careful, you can end up with basically wrong results. Now, with regards to the scattering and this sort of question, what I can say is that for our segmentation work, we often pool together data sets of nuclei that are aberrated or scattered. We find that we can use one sort of mask RCNN for doing many different applications. So that's an example where we can pool data, and we seem to get a more robust, thing where we just keep adding more nuclear examples, and we can get this neural network that we use for many different kinds of microscopes. So, but that's a case where the nuclei actually they're kind of similar, right? I mean, they are nuclei. So I don't have a rigorous answer, but qualitatively, it again sort of seems to boil down to things that look alike to a human, or not.
Yeah, thank you.
Maybe I'll just follow that up with one thing, which is that for some of this de-aberration stuff that I've shown that isn't published yet, we have found kind of empirically we can go fairly far with a physical model where we semi-synthetically aberrate the data, right? And so if we have this physics-based model and we apply that to many different kinds of data, that carries us surprisingly far, even on data that might be aberrated a little bit differently than what we trained on, for what it's worth.
Okay, one last question. I think that we could have a much longer question and answer, but I realize there's a lot of people that also want to see the next presentation. So we'll have one more, and then we'll move on. So the last question here is from Philippe in the UK. Can you concatenate the approaches? Kind of do one thing and then do the next thing. He's talking about improving resolution first and then use less light, or do even more things.
Yeah. So we've done that, and but what we've done is sort of empirical. I mean, we've empirically found that doing this serial thing works much better than trying to have one neural network where you go all the way. And I don't really understand that. I mean, it sort of doesn't make sense to me, but maybe the answer is that empirically, it's a lighter lift, right? If you stack these things. So in some of the later applications that I showed, we denoised, or we deaberrated prior to super resolution. And on some level, it makes sense, right? I mean, you want to correct the aberrations so that you get diffraction limited data before you try and super resolve the data, right? Because it's much easier to do the second thing after the first thing. Then again, if you have ground truth data where you have the answer, why bother, right? And so all I can say is that in what we've tried so far, the serial acquisition, the serial implementation of different neural networks works better than one massive neural network where you try and do it all at once.
Yeah. This is also, for example, GPTL3, for other approaches, so nothing to do with imaging, just to do with text translation. The approach is also a stack of neural networks that have each a very specialized task, instead of trying to train one single network that does everything end to end. Generally, that's the intuition from a lot of people, is to try and do very narrow tasks rather than super complex tasks in one, because it gets actually can confuse the network quite easily if there is too much heterogeneous data. But very much appreciate that. I had one last thing I wanted to ask you, which people often ask or want to know about, is how does this go forward? And there's a few hints towards automation, of creating some sort of loop between the acquisition and then processing and then looping back to control the system. What can you say about this? It's a very exciting, of course, because a lot of biologists like to get the images. They don't like so much to spend a lot of time in the dark room. So that's something exciting for a lot of the biologists. Whereas for some of the other people on the call here, they'll be like, no, that's the fun part. I want to be in the dark room. But I'm wondering, what do you think AI could help in that sense?
Yeah, I mean, I think that's a very promising direction, right? To do smart microscopy where you feed back on the images, and I think for some things like denoising, I mean, if you trust your network, you can already do that on a time scale that is fast enough. For other things, where you're really pushing the envelope of the microscope, and you need the decisions to be made on the millisecond time scale or tens of milliseconds, I think we're not quite there yet, right? I mean, the computer hardware is getting faster all the time, but I think we're not quite there to sort of fully deaberrate data. If you could learn the phase of the wavefront and sort of replace fully replace the kind of wavefront sensor, but I think we could get there, and I think it's a good area to work in because it's kind of underexplored.
Yeah, thank you, Harry, very much for your time and for not only presenting but answering all these questions. I think everybody in the community appreciates your contributions over the years. So it's a pleasure to have you here, and yeah, hope to see you again soon.
Yeah, thanks so much.